Hello, everyone, and welcome to today's webinar. Embrace simple nucleic acid desalting and quantification with Unagi and Lunatic. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Unchained Labs. To learn more, visit unchainedlabs.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use this Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Kevin Lance, Director of Product Management at Unchained Labs. Kevin, you may now begin your presentation.
Thanks for that introduction. I'm excited to talk today about nucleic acids and what Unagi and Lunatic can do for you when it comes to them. Specifically, some buffer exchange and desalting and quantification. If you don't already know, Unagi means eel. So when eels spawn, they have a complex life cycle. They go from Europe or North America to the Sargasso Sea. This is a long journey between freshwater and saltwater and back. And it's so tricky to measure, it's actually only been observed for North American eels once. It's actually this line right here. So why do I bring up this story now? I actually think it's a perfect analogy for DNA and RNA synthesis. It's a long process with a lot of steps. And at the end of it, you want to desalt and be in a nice freshwater buffer. So that's when we get to the idea of buffer exchange. Buffer exchange, when you're working with different kinds of samples, can be essential. So for nucleic acids, the idea is to desalt before other downstream applications after synthesis or purification. And with proteins and antibodies, which we won't really talk about today, but it's definitely something you can do on Unagi. With proteins and antibodies, you can check out different formulations, you can concentrate, or you can just optimize what's going on with your proteins. When it comes to manual methods for buffer exchange, they really don't cut it. Dialysis is slow and your sample ends up diluted, and it can be hard to scale that up to lots of different samples. Centrifugation, well, it runs fast, but you're concentrating your sample at the membrane. As buffer leaves through a membrane, your sample stays behind, and all that happens right at the bottom where your centrifuge is pushing all of your precious sample. That can lead to aggregation and other problems like membrane clogging. And TFF is complicated, requires large sample volume, and again, it's hard to scale up to lots and lots of different molecules at the same time, or just a high number of molecules at the same time. That's why I want to introduce you to our Unagi. It allows you to run up to 8 different exchanges at the same time, using sample volumes between 500 microliters all the way up to 48 milliliters of sample. And you can do that with less than 15 minutes of hands-on time, because Unagi is essentially a liquid handling robot that takes care of all of these buffer exchange steps for you. To handle the buffer exchange or sample concentration or desalting inside of Unagi, samples first go in our consumable, the Una. So each Una is able to handle a 0.5 to 8 milliliter volume range. So if you pipette up to the top, that'll be 8 milliliters of volume. And at the bottom of this Una is a regenerated cellulose or a polyether sulfone membrane in a variety of molecular weight cutoffs. So that's what allows us to push sample through the bottom, have the liquid and salt leave, retain your sample, and then be ready for the next process step. We'll go through this step by step here in the next few slides. Okay, so here's kind of the cutaway of what's going on inside your sample, inside your UNA when it's running on Unagi, which for the technique that we're calling this is an automated pressure-based UF-DUF or ultrafiltration diafiltration step. So we start here with the actual samples in our UNA. Unagi will first use an ultrasonic non-contact probe to measure the volume in an UNA, and then it will apply positive pressure to push sample through the bottom of the membrane. Now what's important here is that we're also mixing these samples so that we're not concentrating the sample at the bottom as liquid leaves. Everything stays really well dispersed throughout the volume of it. After a pressure plus mixing run, we then measure volume again to know what's left in each UNA, and then refill using one of a set of Falcon tubes that you have stored inside the Unagi with things like water or new buffer, whatever you want your sample to end up in, and then repeat that cycle until we reach the desired level of buffer exchange or sample concentration, and that's what's left in the UNA at the end of the cycle. So because we're monitoring the sample volume during an Unagi run, we're able to adapt the lengths of time that UNAs are run with so that you're not going to run your sample dry at the bottom of the UNA, since it's tracking this for every single sample and adapting as it goes. You can also program the pressure level that's in the buffer exchange chamber, and this is how we sort of adapt Unagi runs, usually between different sample types. The other approach that we'll use is how much buffer is removed in each run. So for example, are we removing 50% or removing 33% of the buffer in this cycle here in the middle. Unagi allows you to end up with a sample that's concentrated up to 100x what you started with. The way that we'll do that kind of run is by starting with sample in Falcon tubes. That can be your dilute sample. Unagi will take care of filling or refilling the UNAs during a run of concentration. And then as it applies pressure, it's pushing that extra liquid out through the bottom and adding more dilute sample in and repeating until you end up with your concentrated sample, which is something you can go from 48 mils of total sample up to 500 microliters. This is our reduced sample volume application. We can do this in parallel for up to 8 samples. This is an illustration of that dead end problem that you see with things like centrifugation. On the left, you can imagine you have a filter in a tube and a centrifuge. As the centrifugation runs, you're pushing your small molecules, your diluent, your buffer, through the bottom of your membrane. And what happens is your solutes are left behind, but at a higher concentration right at that membrane, where they're also pushed down to. If you add in Unagi's orbital mixing to the pushing smaller particles through the membrane, you continue to redisperse those large molecules in your sample, and you don't have to worry about those membrane effects where there's very high amounts of particles present at the membrane. This is also something that you can set on Unagi, which would be sort of the mixing rate. RPMs is something you could think about. The mixing rate for how vigorously Unagi is mixing that sample. The idea is that this also, in addition to keeping your sample from being over-concentrated, it also prevents clogging and allows you to run faster buffer exchange or sample volume reduction. All of these applications that I talked about, you get to choose what Unagi is doing with your sample. So you can simply remove a percentage of your sample. You can dictate how much is exchanged between one step to the next, and you can dictate what the final concentration or final sample volume should be for your Unagi run. Okay, so now that we know the Unagi process and we know a little bit more about eels, Unagi, it's time for the next step, which is really, counting how many eels you have, or if we're talking about nucleic acids, quantifying how much DNA and RNA you have. Since it's not just about buffer exchange, it's also about quantification. For quantification, we use Lunatic. Lunatic allows you to run 2 microliters of sample volume for up to 96 samples in a normal 8 by 12 sample plate. And for that whole plate of samples, in about 10 minutes, it's going to give you answers on quantification using UV-Vis. Lunatic's plates are also SBS plate format, so that you can run this on automation and use that for sample loading or for running a whole process with Lunatic. The plate is what makes this all possible. So you see the normal 8 by 12 format here on the left, where each well has actually been replaced with a microfluidic circuit that has an input well where you'd load your 2 microliters of sample volume. That will be held inside of the plate in a microfluidic reservoir. We'll sometimes call that the serpentine path. That protects your sample from any evaporation for several hours. And then inside of Lunatic, the sample will be pumped into these 2 cuvettes, which have fixed path lengths that allow you to do very accurate and precise absorbance readings across the wavelength range of 230 to 750 nanometers. And this is going to give you a readout of the absorbance of your sample volume, which we'll use for quantification. To show what this looks like, this video is in real time, where we added some fluorescein and we're using black light to visualize things. So, Lunatic's not a fluorescence reader, but this really helps us see what happens on video. You can see I added 2 microliters of sample to that well, and it gets wicked into the sample volume just that fast. Inside of Lunatic, samples are pumped into those microcuvettes. So on the left, we have samples that are just loaded and have not been read. And on the right, we have samples that have been pumped into the microcuvettes. They've been read in Lunatic. And you can actually see this is a high Lunatic plate. We're using 2 different path lengths of wells, and that gives us a very large dynamic range. And that's kind of shown by the different brightnesses between those 2 cuvette volumes. That corresponds to the 2 different path lengths, which gives us a huge range of concentrations. The results that you get from this kind of read help you understand the quantification of your DNA and RNA. So what we're looking at here is octuplicate reads of DNA on the left and RNA on the right, and we're measuring the absorbance for each of those N of 8 samples across a dilution series. Now, the precision of Lunatic is such that you can only see maybe in this one case where there's any idea that these are actually 8 lines. We're not actually looking at an average. We're looking at overlaid spectra from 8 different reads across a range of concentrations all the way up to 12,000 nanograms per microliter in the case of DNA. Twofold dilutions across this whole dilution series shows us that we're able to also do a wide range all the way down to about 1.5 nanogram per microliter for double-stranded DNA and plasmids, and down to about 1.2 nanogram per microliter for RNA. The results of this are ridiculously good when it comes to quantification. So you can see here where we have our target concentration and our measured concentration. We have an incredibly linear response with a slope incredibly close to 1, which tells us that our expected value and our measured value agree very well. Our dilution series is very linear. And then you can see by the quantification in the right side of the slide, where we're looking at the average concentration versus target and the percent CV, that we get accuracy within 2% of expected value and precision within 1% of the expected value when we're operating in the range of 1 to 200 OD. When it comes to quantification of nucleic acids, Lunatic is way simpler than other comparable methods. So what's very common out there in the world is using a fluorescence dye, which can be added to a nucleic acid sample, and that requires at least 6 steps to do the additions, to do the dilutions, to be in the right dynamic range, have your standard curve and do your sample read, and then finally all the math afterwards. For Lunatic, that process compares to just having a sample pipetted on your plate, putting it into the software, and getting a result out. So there's no reagents, no dilutions, no complicated workflow on Lunatic. It just simply runs the sample and gets you a result of quantification that you can move on with. Importantly, once you start looking at the cost of these 2 processes, then having that reagent really starts to add up when you're running lots and lots of samples for your nucleic acid quantification. By consumable costs alone, It far exceeds the cost of running on Lunatic. And once you add up the labor cost savings involved in that multi-step process versus what's involved in Lunatic, then you can very quickly actually have a cost savings beyond the cost of a Lunatic when you're running a lot of nucleic acid quantification samples. Okay, so let's get back to how we can use Unagi and get to the Unagi of our dreams and what we're trying to actually get to with our nucleic acid process. What's inside of Unagi is a buffer rack of samples, a buffer exchange chamber where we're going to do that positive pressure and run our nucleic acid samples, that UNA sample rack with 8 UNAs. Unagi is able to pipette samples with a pipette channel and also has an ultrasonic volume sensor. And then we're, of course, using multiple tips. So you have tip rack and waste bin inside. Each well does get its own volume measurement. So here you can see the ultrasonic volume sensor checking the edges of the UNA and checking the top level of that sample, so it understands how much sample is present, whether that's before or after a buffer exchange run. Unagi will handle the buffer exchange addition for you. So here we can see it's pulling up some nice blue buffer and adding it into an UNA where it's getting ready for another round of positive pressure, pushing that buffer through the sample. And then finally, once all of the sample volume measurements are done and the buffer is loaded, we get orbital mixing inside of the buffer exchange chamber that's sealed that allows the positive pressure to push the fluids down through the bottom of the UNA. The results of this, the setup for this varies between different sample types. So we're going to adjust pressure and adjust target removal percentages at various presets that we've already optimized. So for your sample type, you can walk up and run with settings that are ideal for handling whatever you want to buffer exchange or concentrate. If we're working with, for example, proteins, then our buffer exchange steps are optimized here for buffer exchange with concentrate, or reduce sample volume, where we see we're getting concentrations that in some cases are exactly what we started with, in some cases have been up concentrated, and a recovery that's really close to 100%, so in this case above 96% for each case. And then in the case of reduced sample volume, we've gone from 48 milliliters down to a volume that we actually want to work with. I always love talking about those kinds of concentrate or reduced sample volume applications because I always imagine being in lab and diluting something too much and going, oh no, what do I do now? Unagi can actually solve that problem. If we look at other sample types, so here we're back to DNA, we can see that our recovery is still at 95% or better. We want to study a use case for DNA. Here we're looking at hands-free desalting of a salmon sperm DNA that is about 2 kilobase in length at a concentration about 2 mg per mil. And what we wanted to do with Unagi was simply exchange that double-stranded DNA into TE buffer, or that was in TE buffer into nuclease-free water. At the same time, we also wanted to concentrate this threefold, and we're going to use a 30 kilodalton UNA in this case. After running that sample on Unagi, we know our sample is cleaned up and concentrated in one go. In this case, we're using the nucleic acid preset, where I showed the parameters for that on an earlier slide. But the DNA parameters, what we've seen in our sample, we start with our concentration around 2.1 mg per mil. We are achieving our threefold concentration between our initial and final sample. If we're measuring the volume that we input into Unagi and the volume that we're getting out after that concentration run, we are also seeing that volume reduction that corresponds to that threefold concentration step. And then our recovery and percent exchange is greater than 96%, so we're being very efficient with our sample at the same time. So we've achieved our concentration we want, and we've not lost sample unnecessarily. If we want to confirm that our buffer exchange has happened, here we have an example where we're looking at acetonitrile that's present in our buffer beforehand, since it's commonly used in DNA synthesis. An easy way to track acetonitrile content versus water is by looking at the refractive index. So you can see kind of on this standard curve for a sample that we're running, we have the relationship between refractive index and acetonitrile. In this case, we're looking at a sample that started with a refractive index close to 1.34, just a little bit below it because we were starting at 15% acetonitrile. Using our UNAs, we were actually able to reduce that all the way down to levels where the refractive index was 1.33, which 1.333, I should say, there's an extra 3 in there, which correspond to less than 1% of acetonitrile remaining in the sample. And it also demonstrated to us beyond that we could remove the acetonitrile. It demonstrated us that we were compatible with acetonitrile with our UNA materials. Lastly, I want to show you quickly a little bit about Unagi's software and doing this whole process. It's set up to make all of these processes very easy with a user-friendly wizard and a lot of presets that enables you to get your molecule in the buffer or concentration that you want. It walks you through setup, so that Unagi platform that we saw earlier, you can know exactly what to add and where on the deck. And it helps you stay in the loop as this process runs, because you can see a status as it's progressing on the software, but you can also receive text messages alerts for what Unagi is up to on your phone. Out of Unagi, we're also going to get a report of everything that's been done and the results for it. That's reported for individual samples. That's reported for the experimental parameters overall. And also, we can dig in and get the raw report of what's going on for under the hood for everything that Unagi is up to. All of that shows us that Unagi enables us to do fast and flexible buffer exchange or desalting with nucleic acids, gives us a lot of control over that process in a way that allows us to set up the sample and then have low hands-on time after that and go be productive in lab. It gives us results with high recovery on our samples and high quality at the end of it, and also has multiple membrane options to be sure that we're picking the right one for the task that we have in mind. And at the end of the day, you can get to the Unagi that you really want, which is a nice, beautiful, cleaned up sample. That's all for my conversation about Unagi and Lunatic and what they can do for DNA desalting and quantification. Now, I'd love to take any questions you have for me.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's get started. Our first question asks, you showed a quick cost comparison. What was the cost per sample for Lunatic?
Oh yeah, so on that graph, I was comparing Lunatic versus dye-based fluorescence assay, usually picograins used for that. So Lunatic runs about 40 cents per read, so that's 40 cents per well. Since there's no dyes, reagents, or standards needed, that's the total cost per sample, all in, and that's going to be significantly less than using a fluorescence dye-based method.
Excellent. All right, moving on to our next question here. This next question asks, what other kinds of samples can I quantify with Lunatic?
We showed some data for some kind of generic DNA samples, calf, thymus, and salmon sperm DNA, but typically customers are reading oligos, genomic DNA, plasmids, RNA, and Lunatic can actually handle other things as well. So things like proteins, antibodies, peptides, even colorimetric assays and small molecules too. So anything that absorbs in the UV vis between 230 and 750, Lunatic can handle it.
Great, thank you. All right, our next question here. What are the MWCO options available for Unagi's consumables?
Sure. So Unagi has a variety of different molecular weight cutoffs, MWCOs. So that's regenerated cellulose, where we have that available in 10, 30, and 100 kilodalton molecular weight cutoffs, and polyether sulfone is available in 30 and 100 kilodalton molecular weight cutoffs. So we kind of try to choose those so they would work for a range of customer applications.
Great, thank you. These are excellent questions. Keep them coming. Please submit them. The Ask a Question box is in the lower left corner. Our next question here, what is Lunatic's dynamic range and can it get down into the single nanogram per microiter?
So Lunatic's dynamic range in OD, which is usually the language of instruments, is between 0.03 and 275 OD, an equivalent 1 centimeter cuvette. So what does that really mean? For DNA samples, that's between about 1.5 nanogram per microliter, all the way up to more than 13,000 nanogram per microliter. So, and then for RNA, it's down to 1.2 nanogram per microliter on the low end. So yeah, we can get down into the single-digit microliter concentrations, which is usually plenty of dynamic range to quantify a lot of these nucleic acid samples.
Wonderful, thank you. Okay, our next question here. In Unagi, why are there 16 tubes for buffer, but only 8 samples you can run?
So sharp eyes. So for some kinds of runs, you might require more than 48 milliliters of buffer or sample if you're doing a reduced sample volume operation. So that gives you more volume to run through a single UNA. The two 50 mil Falcon tubes are associated with one UNA. So we allow a 2-to-1 ratio of buffer tubes and UNAs.
Okay, excellent. Next question here. You mentioned a lower volume limit of 0.5 ml. Can you start at that level and concentrate down?
So our answer is no. That's the lower end of the working volume range. So we use that lower limit to minimize the effects of dead volumes and any other problems you might have where a sample gets over-concentrated at the membrane, since the whole point of Unagi is to keep everything well dispersed in the buffer as that buffer exchange is going on.
Excellent, thank you. All right, our next question here asks, what are the factors that impact the length of buffer exchange?
Okay, so there's a lot. There's the sample itself. There's the buffer itself. Those are two that we don't have control over, and it definitely depends on sample to sample. What you can control inside of Unagi are the pressure that's applied, the RPMs that's applied. So the samples are moving, rotate, or I guess RPMing while you're doing buffer exchange, and then the percentage of buffer exchange. So for each run, do you want to remove 50%, 33%, 10% of your buffer down through the bottom of the membrane? And if you can imagine, if you're moving 10% every cycle, you're going to need more cycles to get to complete buffer exchange than you would if you're moving 50% every cycle. And those will all be factored into the duration of a sample run.
All right, excellent. And it looks like we have one more question here today. So this last one is asking, how long can samples be held in the SBS plate prior to read on the Lunatic instrument?
So about 2 hours, and that's before you're putting it into the Lunatic. That's our typical guidance. Usually that comes in handy when you're talking about, say, loading a lot of samples, maybe in an automation setup prior to reading. But the idea is that small sample volume is secured within the Lunatic plate and kind of sheltered from evaporation during those 2 hours.
All right, excellent. I want to thank you for your time today and your important research. We would also like to thank LabRoots and our sponsor, Unchained Labs, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.